The GMO versus gene editing debate: who will win the battle for public acceptance? This article provides a summary of its contents in the following manner. 1. Introduction, explanation of GMOs and gene-edited foods, brief overview of the differences between the two. 2. GMOs, definition and explanation of GMOs, the process of creating GMOs, advantages and disadvantages of GMOs, controversies and concerns surrounding GMOs, regulatory oversight of GMOs. 3. Gene-edited foods, definition and explanation of gene-edited foods, the process of creating gene-edited foods, advantages and disadvantages of gene editing, controversies and concerns surrounding gene editing, regulatory oversight of gene-edited foods. 4. Comparison of GMOs and gene-edited foods, similarities and differences between GMOs and gene-edited foods, potential benefits and drawbacks of each approach, public perception and acceptance of GMOs versus gene-edited foods. 5. Future of genetic engineering in agriculture, potential advancements in genetic engineering technology, potential future uses and applications of GMOs and gene-edited foods, ethical and societal considerations for the future of genetic engineering in agriculture. 6. Conclusion, final thoughts on the debate between GMOs and gene-edited foods, call to action for further research and discussion. 1. Introduction. GMOs, genetically modified organisms, and gene-edited foods are two types of genetically engineered crops that are created using different techniques. GMOs are created by introducing foreign DNA into the genome of an organism in order to confer a desired trait, such as resistance to pests or herbicides. This foreign DNA is typically created using recombinant DNA technology, where a gene from one organism is isolated and then inserted into the genome of another organism. The resulting GMO may have genetic material from multiple different organisms. Gene-edited foods, on the other hand, involve precise changes to an organism's existing DNA sequence using tools such as CRISPR-Cas9. With gene editing, specific genes can be targeted and modified without the introduction of foreign DNA. This allows for more precise genetic engineering without the potential risks associated with introducing foreign genetic material. In terms of the resulting products, both GMOs and gene-edited foods can have similar traits, such as improved yield, resistance to pests or disease, and enhanced nutritional content. However, the processes used to create them and the resulting products may differ in terms of potential risks, regulatory oversight, and public perception. The main difference between GMOs and gene-edited foods is in the way they are created. GMOs involve the introduction of foreign DNA into the genome of an organism to confer a desired trait, while gene-edited foods involve precise editing of existing genes within an organism's genome. This difference has several implications. For example, gene editing does not necessarily involve the introduction of foreign genetic material, which can reduce concerns around safety and environmental impacts. Additionally, GMOs are subject to more regulatory oversight than gene-edited foods due to the use of foreign DNA. Another difference is that gene editing can be more precise than GMOs, as it allows for the modification of specific genes without affecting others. GMOs, on the other hand, can result in the expression of unintended traits due to the insertion of foreign genetic material. Overall, both GMOs and gene-edited foods have the potential to offer benefits such as increased crop yields and improved resistance to pests and disease, but also raise questions about safety, ethical concerns, and potential impacts on the environment. 2. GMOs. GMOs, genetically modified organisms, are organisms whose genetic material has been altered in a way that does not occur naturally through mating or natural recombination. In the context of agriculture, GMOs typically refer to crops that have been genetically engineered to have specific traits such as resistance to pests or herbicides. The process of creating GMOs typically involves the use of recombinant DNA technology. This involves the isolation of a desired gene from one organism and then inserting it into the genome of another organism. The resulting GMO may have genetic material from multiple different organisms, and the introduced gene can confer a desired trait such as pest resistance. Some advantages of GMOs include increased crop yields, improved nutritional content, and increased resistance to pests and disease. However, there are also concerns around the safety and environmental impact of GMOs, particularly with regard to the potential introduction of foreign genetic material into the ecosystem. GMOs are subject to regulatory oversight in many countries, with varying levels of stringency depending on the country and the specific GMO in question. Despite ongoing debates and controversies, GMOs continue to be used in agriculture around the world. The process of creating GMOs typically involves the use of recombinant DNA technology. Here is a general overview of the steps involved. Isolation of the gene of interest. The first step in creating a GMO is to identify and isolate the gene that encodes the desired trait. This may involve searching for the gene in other organisms, or identifying it within the genome of the target organism itself. Cloning of the gene. Once the gene of interest has been identified, it is amplified through a process called PCR, polymerase chain reaction, to produce multiple copies of the gene. Insertion of the gene into a vector. The gene of interest is then inserted into a vector, which is a small DNA molecule that can be used to transfer genes into cells. The vector is typically engineered to include sequences that will ensure that the gene is expressed in the target organism. Transformation of the target organism. The vector containing the gene of interest is then introduced into the cells of the target organism, which may be a plant, animal, or microorganism. This can be done using a variety of techniques such as electroporation, particle bombardment, or bacterial-mediated transformation. Selection of transformed cells. Once the vector has been introduced into the cells of the target organism, the transformed cells are screened to identify those that have successfully taken up the vector and incorporated the gene of interest into their genome. Propagation and testing of the GMO. The cells that have successfully integrated the gene of interest are then propagated to produce a population of GMOs. These GMOs are then tested to ensure that they exhibit the desired trait, and to assess their safety and environmental impact. The process of creating a GMO can be complex and time-consuming, and may involve the use of specialized equipment and techniques. The resulting GMOs may offer benefits such as increased crop yields and improved resistance to pests and disease, but also raise concerns around safety and environmental impact. Advantages of GMOs. Increased crop yields. GMOs can be engineered to have increased resistance to pests, disease, and environmental stressors, which can result in higher crop yields. Improved nutritional content. GMOs can be engineered to have improved nutritional content, such as higher levels of vitamins or essential amino acids. Reduced need for pesticides. GMOs can be engineered to have built-in resistance to pests or herbicides, which can reduce the need for chemical pesticides and herbicides. Longer shelf life. GMOs can be engineered to have longer shelf lives, which can reduce food waste and increase food availability. Disadvantages of GMOs. Potential environmental impacts. GMOs have the potential to negatively impact the environment by introducing foreign genetic material into ecosystems, disrupting natural food chains, and reducing biodiversity. Safety concerns. Some people have concerns around the safety of consuming GMOs, although extensive research has not found any significant health risks associated with GMO consumption. Ethical concerns. Some people have ethical concerns around the use of GMOs, particularly with regard to the ownership of genetic material and the potential for corporate control of the food supply. Regulatory issues. There is ongoing debate and controversy around the regulation of GMOs, particularly with regard to labeling and transparency. While GMOs offer potential benefits such as increased crop yields and improved nutritional content, there are also concerns around safety, environmental impact, and ethical considerations that need to be carefully considered. GMOs have been the subject of significant controversy and concern since they were first introduced in the 1990s. Some of the main controversies and concerns surrounding GMOs include environmental impact. One of the main concerns around GMOs is their potential environmental impact. Critics argue that the introduction of foreign genetic material into ecosystems could have unintended consequences, such as disrupting natural food chains and reducing biodiversity. Human health. Some people are concerned that consuming GMOs could have negative health effects. However, extensive research has not found any significant health risks associated with GMO consumption. Corporate control. Some people have raised ethical concerns around the use of GMOs, particularly with regard to the ownership of genetic material and the potential for corporate control of the food supply. Regulation. The regulation of GMOs has been a contentious issue, with some people arguing that GMOs are not adequately regulated and that there is a lack of transparency around the use of GMOs in the food supply. Labeling. There has been significant debate around the labeling of GMOs, with some people arguing that consumers have a right to know whether the food they are consuming contains GMOs. Cross-contamination. There are concerns around the potential for cross-contamination between GMO crops and non-GMO crops, which could result in unintended exposure to GMOs. While there are certainly valid concerns around the use of GMOs, it is important to note that GMOs have been extensively studied and evaluated for safety, and have been found to be safe for consumption by humans and animals. However, ongoing debates and controversies around issues such as labeling and regulation suggest that GMOs will continue to be a contentious issue for the foreseeable future. Regulatory oversight of GMOs varies by country, but in general, most countries have regulatory bodies that oversee the development, testing, and commercialization of GMOs. Here is an overview of how GMOs are regulated in the United States. The development of a GMO typically begins with an application to the U.S. Department of Agriculture, USDA, which oversees the development and introduction of GMO crops. The application process involves a review of the potential environmental impact of the GMO, as well as an assessment of its potential to cause harm to non-target organisms or ecosystems. If the USDA determines that the GMO is safe, the developer can then proceed to field testing. The developer must also submit an application to the Environmental Protection Agency, EPA, if the GMO contains a pesticide or if it is intended to have a pesticidal effect. The EPA review process includes an assessment of the potential risks to human health and the environment, as well as an evaluation of the effectiveness of the pesticide. Finally, the developer must also submit an application to the Food and Drug Administration, FDA, if the GMO is intended for human or animal consumption. The FDA review process involves an evaluation of the safety and nutritional content of the GMO, as well as an assessment of its potential to cause allergenic reactions. The regulatory oversight of GMOs in the United States is carried out by multiple agencies, with each agency responsible for evaluating different aspects of the GMO. The goal of this regulatory oversight is to ensure that GMOs are safe for human and animal consumption, and do not pose significant risks to the environment. 3. Gene-edited foods. Gene-edited foods are foods that have been produced using a new set of techniques known as gene editing, which involves making precise, targeted changes to an organism's DNA. Gene editing differs from traditional genetic engineering, which involves transferring genes from one organism to another, often from different species. Gene editing technologies, such as CRISPR-Cas9, allow scientists to make specific and precise changes to an organism's DNA, such as turning off a particular gene or modifying an existing gene sequence. This can be used to create crops that are more resistant to pests and diseases, that have improved nutritional content, or that are better suited to specific growing conditions. One of the key benefits of gene editing is that it allows scientists to make precise changes to an organism's DNA without introducing genes from other organisms. This can help to alleviate some of the concerns around traditional genetic engineering, such as the potential for unintended consequences or for the transfer of allergenic or toxic genes. Gene-edited foods are still relatively new, and there is ongoing debate around their safety and regulation. However, early research suggests that gene-edited foods have the potential to offer significant benefits, such as increased crop yields, improved nutritional content, and reduced environmental impact. The process of creating gene-edited foods involves several steps. Identify the target gene. Scientists start by identifying the specific gene that they want to edit in the target organism, such as a crop plant. Design the gene editing tool. Once the target gene has been identified, scientists then design a gene editing tool that can be used to make precise changes to the DNA. The most commonly used tool is CRISPR-Cas9, which allows scientists to cut and paste specific sections of DNA. Edit the DNA. Using the gene editing tool, scientists then make precise changes to the target gene in the organism's DNA. This can involve deleting or adding specific sections of DNA, or making smaller changes to the gene sequence. Screen the edited organisms. After the gene editing is complete, scientists then screen the edited organisms to identify those that have the desired genetic traits. This can involve selecting for specific traits such as increased yield or improved disease resistance. Test for safety. Before the edited organism can be used in food production, it must undergo rigorous safety testing to ensure that it does not pose any risks to human health or the environment. Regulatory approval. Once the edited organism has been deemed safe, it must then undergo regulatory approval by government agencies such as the FDA in the United States or the European Food Safety Authority in Europe. The regulatory process typically involves a review of the safety and potential environmental impact of the edited organism. The process of creating gene-edited foods is similar to that of traditional genetic engineering, but with the added precision and control of gene editing tools such as CRISPR-Cas9. The goal is to create crops that have desirable genetic traits and that are safe for consumption by humans and animals. Gene editing has the potential to offer several advantages, but there are also potential disadvantages that need to be considered. Here are some of the main advantages and disadvantages of gene editing. Advantages. Precise changes. Gene editing allows scientists to make precise changes to an organism's DNA, which can help to create crops that have specific traits such as increased yield, improved disease resistance, or enhanced nutritional content. Increased efficiency. Gene editing is often faster and more efficient than traditional breeding methods, which can take many years to develop new crop varieties. Reduced environmental impact. By creating crops that are more resistant to pests and diseases, gene editing can help to reduce the need for harmful pesticides and herbicides. Improved food security. Gene editing can help to create crops that are better suited to specific growing conditions, which can improve food security in regions where traditional crops may not thrive. Disadvantages. Potential risks. There is still much that is unknown about the long-term risks and unintended consequences of gene editing, both in terms of human health and the environment. Ethical concerns. Gene editing raises ethical concerns around the use of biotechnology to alter the fundamental nature of living organisms. Unequal access. The development of gene-edited crops is often expensive, which could limit access to these technologies for farmers in low-income countries. Regulatory challenges. The regulatory oversight of gene editing is still in its early stages, and there is a need for clear and consistent regulations to ensure the safety and ethical use of these technologies. Gene editing has the potential to offer significant benefits in terms of crop development, but there are also important ethical and safety concerns that need to be carefully considered. Gene editing is a relatively new and rapidly developing field, and as such, there are several controversies and concerns that surround its use. Here are some of the main controversies and concerns surrounding gene editing. Safety concerns. One of the main concerns around gene editing is the potential for unintended consequences or unexpected effects. While gene editing tools such as CRISPR-Cas9 are precise, there is still a risk that unintended changes could occur in the organism's DNA that could have negative consequences for human health or the environment. Ethical concerns. Gene editing raises important ethical questions around the use of biotechnology to alter the fundamental nature of living organisms. There are concerns around the potential for gene editing to be used to create designer babies or to enhance human traits beyond what is considered normal. Unequal access. The development of gene-edited crops is often expensive, which could limit access to these technologies for farmers in low-income countries. This could exacerbate existing inequalities in the global food system. Regulatory challenges. The regulatory oversight of gene editing is still in its early stages, and there is a need for clear and consistent regulations to ensure the safety and ethical use of these technologies. However, there is currently no international consensus on how to regulate gene editing, which could lead to confusion and inconsistencies in regulation across different countries and regions. Public perception. There is still a lack of public awareness and understanding around gene editing, which could lead to mistrust and resistance to the development and use of gene-edited crops. While gene editing offers significant potential benefits, there are important safety, ethical, and regulatory concerns that need to be carefully considered and addressed. The regulatory oversight of gene-edited foods varies depending on the country or region. In general, gene-edited crops are subject to similar regulations as GMOs, although some countries have begun to differentiate between the two. In the United States, the regulation of gene-edited crops falls under the purview of the United States Department of Agriculture, USDA, and the Food and Drug Administration, FDA. The USDA regulates gene-edited crops that have been developed using plant pests or that could pose a risk to the environment, while the FDA regulates gene-edited crops that are intended for human or animal consumption. The Environmental Protection Agency, EPA, also plays a role in the regulation of gene-edited crops, as it oversees the use of pesticides and herbicides that may be used in conjunction with these crops. In the European Union, EU, gene-edited crops are regulated under the same laws as GMOs. This means that gene-edited crops must undergo a rigorous approval process before they can be marketed and sold within the EU. However, there is currently some debate around whether gene-edited crops should be subject to the same regulations as GMOs, as some argue that the precision of gene editing techniques means that these crops pose less risk to the environment and human health. In other countries such as Japan, Canada, and Australia, Regulatory oversight of gene-edited crops also falls under the same regulations as GMOs. The regulation of gene-edited foods is still evolving, and there is ongoing debate around how these crops should be regulated and what kind of safety and environmental standards they should be held to. 4. Comparison of GMOs and gene-edited foods. GMOs and gene-edited foods are similar in that they both involve the genetic modification of organisms, but there are also important differences between the two. Process: GMOs are created through the insertion of genes from one organism into another using biotechnology techniques. Gene-edited foods, on the other hand, are created through precise edits to an organism's existing genes using tools such as CRISPR-Cas9. Precision: while GMOs involve the insertion of new genes into an organism's genome, gene editing allows for more precise modifications to an organism's existing genes, making it a more targeted and precise method of genetic modification. Regulatory oversight: in most countries, GMOs are subject to more stringent regulations than gene-edited foods. This is because gene-edited foods are often considered to be more precise and have fewer unintended effects on the environment and human health than GMOs. Public perception: GMOs have been a controversial topic for many years, with many people expressing concerns about their safety and potential impact on the environment. Gene-edited foods are still relatively new, and public opinion around their safety and acceptability is still developing. Applications: while both GMOs and gene-edited foods have potential applications in agriculture, gene editing is also being explored for use in medicine, with the potential to develop new treatments and cures for diseases. While both GMOs and gene-edited foods involve the genetic modification of organisms, gene editing offers a more precise and targeted approach to genetic modification that has the potential to address some of the concerns around the safety and environmental impact of GMOs. Potential benefits and drawbacks of GMOs. Benefits: increased crop yields and resistance to pests and diseases can help to ensure global food security and reduce food prices. Enhanced nutritional value of crops can help to address malnutrition in developing countries. Reduced use of pesticides and herbicides can lead to lower environmental impact and improve the health of farmers and surrounding communities. Improved shelf life of crops can reduce food waste and increase availability of fresh produce. Drawbacks: potential risks to the environment, including the development of superweeds and the unintended harm to non-target organisms. Potential risks to human health, such as allergies or toxicity. Potential ethical concerns around the patenting of living organisms and the impact on biodiversity. Public perception concerns and potential consumer rejection of GMO products. Potential benefits and drawbacks of gene-edited foods. Benefits: precision editing allows for targeted genetic modifications that can improve the nutritional value, flavor, and texture of foods. Gene editing can reduce the need for harmful chemicals and pesticides in agriculture, leading to a more sustainable food system. Gene editing can help to address the challenges of climate change by creating crops that are more resistant to droughts and extreme weather conditions. Gene editing has the potential to develop new treatments and cures for genetic diseases in humans. Drawbacks: the long-term effects of gene-edited organisms on the environment and human health are not fully understood. Regulatory oversight of gene-edited organisms is still developing and there is uncertainty around safety standards. There are potential ethical concerns around the use of gene editing, particularly in humans. Public perception concerns and potential consumer rejection of gene-edited products. While both GMOs and gene-edited foods have potential benefits in terms of improving food security and addressing nutritional deficiencies, they also carry potential risks and drawbacks that need to be carefully considered and managed. Public perception and acceptance of GMOs and gene-edited foods differ based on several factors, including cultural norms, scientific literacy, media coverage, and public education efforts. GMOs have faced considerable public skepticism and opposition, with many consumers expressing concerns about the safety, environmental impact, and ethical implications of genetic modification. This has led to labeling regulations in many countries, consumer boycotts, and calls for stricter regulations and oversight of GMOs. Some studies have also found that consumers are willing to pay more for non-GMO or organic products, indicating a preference for alternatives to genetically modified foods. In contrast, gene-edited foods are still relatively new and less well known among the general public. Some studies have shown that consumers may be more accepting of gene-edited foods than GMOs, as they are seen as more precise and natural. However, other studies have found that there is still significant public concern around gene editing, particularly in the context of human applications, such as gene editing for disease prevention or enhancement. Public perception and acceptance of GMOs and gene-edited foods are complex and influenced by a range of factors. It is important for regulators, scientists, and food producers to engage with the public, provide transparent information, and address concerns in order to foster a more informed and positive public perception of these technologies. 5. Future of genetic engineering in agriculture. Advancements in genetic engineering technology are continuing to evolve rapidly, opening up new possibilities for improving crop yields, enhancing nutritional content, and developing new medical treatments. Some potential advancements in genetic engineering technology include CRISPR-Cas9. This revolutionary gene editing tool has made it easier and cheaper to precisely modify genes in a wide variety of organisms. It has the potential to be used to develop crops that are more resistant to pests, diseases, and environmental stressors, as well as new treatments for genetic diseases. Synthetic biology. This field combines engineering and biology to create new biological systems, molecules, and devices that don't exist in nature. Synthetic biology could be used to create new plant varieties that are more resistant to disease or have enhanced nutritional content. Gene drives. This technology allows scientists to genetically modify organisms in a way that makes it more likely for the modified genes to be passed on to future generations. Gene drives could be used to control populations of disease-carrying insects or invasive species. Genetic circuits. These are engineered systems that allow genes to be turned on or off in response to specific environmental or cellular signals. This technology could be used to develop crops that respond to changes in soil moisture or nutrient levels. Genome sequencing and analysis. Advancements in sequencing technology and bioinformatics are allowing scientists to rapidly sequence and analyze the genomes of a wide variety of organisms. This information can be used to identify genes responsible for desirable traits, understand the mechanisms of genetic diseases, and develop new treatments and therapies. These advancements in genetic engineering technology have the potential to revolutionize agriculture, medicine, and biotechnology. However, it is important that they are developed and deployed in a responsible and transparent manner, taking into account ethical, social, and environmental considerations. GMOs and gene-edited foods have the potential to revolutionize agriculture, food production, and health. Some potential future uses and applications of these technologies include improved crop yields. By introducing genes that make crops more resistant to pests, diseases, and environmental stressors, GMOs and gene-edited crops could help improve crop yields and food security. Enhanced nutritional content. Genetic engineering could be used to develop crops with higher levels of essential vitamins and minerals, improving the nutritional quality of the food supply. Reduced use of pesticides and herbicides. GMOs and gene-edited crops could be engineered to be more resistant to pests and weeds, reducing the need for chemical pesticides and herbicides. Disease resistance. Genetic engineering could be used to develop crops that are more resistant to diseases and pests, reducing crop losses and improving food security. New pharmaceuticals. Gene-edited plants and animals could be used to produce new drugs and therapies, including vaccines, cancer treatments, and antibiotics. Food waste reduction. Genetic engineering could be used to develop crops that have longer shelf lives, reducing food waste and improving food availability. Environmental sustainability. GMOs and gene-edited crops could be engineered to be more environmentally sustainable, such as crops that require less water or are better adapted to marginal lands. Food allergy prevention. Genetic engineering could be used to develop crops that are hypoallergenic, reducing the risk of food allergies. However, it is important to note that many of these potential applications are still in the research and development phase and will require significant testing, regulation, and public acceptance before they can be widely adopted. Additionally, there are concerns around the potential unintended consequences and ethical implications of genetic engineering, which must be carefully considered and addressed. The future of genetic engineering in agriculture raises important ethical and societal considerations that must be carefully considered and addressed. Some of the key considerations include environmental impacts. Genetic engineering could have unintended environmental consequences, such as the development of pesticide-resistant pests or the transfer of engineered genes to non-target species. It is important to carefully evaluate and regulate the potential environmental impacts of genetically engineered crops and organisms. Food safety. GMOs and gene-edited foods must undergo rigorous safety testing to ensure they are safe for human consumption. It is also important to consider the potential long-term health effects of consuming genetically modified foods. Equity and social justice. The benefits of genetic engineering should be distributed equitably and not disproportionately benefit certain groups at the expense of others. Additionally, the use of genetic engineering in agriculture should not exacerbate existing social inequalities or contribute to the marginalization of certain communities. Intellectual property and ownership. There are concerns that the intellectual property and ownership of genetically engineered crops and organisms could be concentrated in the hands of a few large corporations, leading to monopolies and limiting access for smaller farmers and communities. Public trust and transparency. The development and deployment of genetically engineered crops and organisms must be transparent and involve public consultation and engagement. Building public trust in genetic engineering requires transparency, clear communication, and collaboration with a range of stakeholders. Ethical implications. Genetic engineering raises important ethical questions, such as the use of animal models for research, the potential for unintended consequences, and the balance of benefits and risks. It is important to carefully consider the ethical implications of genetic engineering in agriculture and ensure that these technologies are developed and used in an ethical and responsible manner. Genetic engineering has the potential to revolutionize agriculture and improve food security, but it is important to carefully consider the ethical and societal implications of these technologies and ensure that they are developed and used in a responsible and transparent manner. 6. Conclusion. The debate between GMOs and gene-edited foods is a complex and ongoing one. Both technologies have the potential to revolutionize agriculture and address important global challenges, such as food security, environmental sustainability, and public health. However, each technology also has its own advantages and disadvantages, and there are valid concerns and controversies around both GMOs and gene editing. It is important to carefully evaluate and regulate the development and deployment of these technologies to ensure that they are safe, effective, and beneficial for society as a whole. This requires transparent communication and collaboration with a range of stakeholders, including scientists, policymakers, farmers, and consumers. Ultimately, the goal should be to harness the potential of genetic engineering to create a more sustainable, equitable, and resilient food system, while also respecting ethical and societal considerations. There is still much to learn and understand about the potential benefits and drawbacks of genetic engineering in agriculture, and ongoing research and discussion is critical. For example, research is needed to better understand the long-term environmental impacts of GMOs and gene-edited crops, as well as the potential unintended consequences of gene editing. We also need more research to assess the safety and nutritional value of GMOs and gene-edited foods, and to address concerns around potential allergenicity and other health risks. In addition to scientific research, it is important to engage in ongoing discussions and debates about the ethical and societal implications of genetic engineering in agriculture. This includes conversations around issues such as intellectual property and ownership, equity and social justice, and public trust and transparency. Continued research and discussion is essential to ensure that genetic engineering is deployed in a responsible and sustainable way, and that the benefits of these technologies are realized while minimizing potential risks and harms. Genetic engineering in agriculture, including both GMOs and gene-edited foods, has the potential to offer significant benefits to society, including improved crop yields, enhanced nutritional content, disease resistance, and reduced use of pesticides and herbicides. However, there are also valid concerns and controversies around both technologies, including potential environmental impacts, food safety, equity and social justice, intellectual property, and ethical implications. Regulatory oversight of GMOs and gene-edited foods is important, as is ongoing scientific research and discussion to better understand the potential benefits and drawbacks of these technologies. Ultimately, the goal should be to harness the potential of genetic engineering to create a more sustainable, equitable, and resilient food system, while also respecting ethical and societal considerations. It is important to continue to engage in open, transparent, and collaborative discussions around the future of genetic engineering in agriculture, involving a range of stakeholders, including scientists, policymakers, farmers, and consumers. By doing so, we can work towards a future where the benefits of genetic engineering are realized in a responsible and sustainable way. Thanks for watching.